Good evening to all. First, I will introduce myself. I am as a Malati Salunke, as a assistant professor in the Pune College of Pharmacy, RN1 Pune. Today, we will discuss on the topic as a history of the plant tissue culture. First, we will see what is meant by the plant tissue culture. Simply, the plant tissue culture is refers the techniques of the growing plant cells, tissues, organs, seeds, and other plants parts in the sterile environment on the nutrient medium using the in vitro conditions only. Uh, then main history under the plant tissue culture is that the Haberland realized that the asepsis was necessary when culture media enriched with organic substances metabolized by the microorganisms. In 1998, he cultured the single cell of a no salt solution simply. Then after that, his culture the cell were able to synthesize the starch as well as increase in size. Then he failed in his goal to induce the cell division. But after that, the despite of the failures, Haberland proposed that plant cells are the totipotent in nature. First, he defined by itself. The first scientist for recognizing plant cells are the totipotency in their nature is the Haberland only. The totipotency is the ability of individual cells to express the phenotype of a whole plant from which it is derived. Then that Haberland is also regarded as a father of a plant tissue culture. Is it clear that scientist that introduced the totipotency of the plant cells as a Haberland, and it is also called as the father of the plant tissue culture. Then next one is a Hanning embryo culture of the selected crucifers that can be work on the that embryo culture. Okay. Then Robbins as a in vitro culture of the root tips. They work on the root tips. Okay. Lebach in 1925 he worked on the embryo culture techniques. He introduced the firstly that using the embryo we can be study under the PTC in interspecific crosses of the linum. And then in the history, next one is a White, the scientist as White in the 1934-37, successful culture of the tomato roots, importance of the vitamins in the growth media. That's no. The next scientist in the 1934 to 39 as a Snow and Gutheret, that the importance of the auxins as a growth regulator hormones can be introduced by firstly by the Snow and the Gutheret in the study of the itself. Then Gutheret and the Novgorod and the White, these three scientists in the 1935 successfully established establishment of the continuously growing culture media for containing the different nutrients. Then next one is a Van Overbeek in the 1941 used the coconut oil firstly for containing a cell division factors first time in a Dhatura tissues. Okay. Here they introduced as a coconut milk medium for the growing cells in a minimum cost they can be attain the end of his study using the on the Dhatura plant study. The next one is the Skoog and the Susi in the 1951 and the Miller in the 1955. In vitro cell differentiation can be studied discovery of the kinetin. Okay. The next one is the Morel and the Martin 1952 applications of the micrografting to obtain the virus-free plants. Then next scientist as a more in the 1953 he can study on the isolation and culture of the single cell from the plants. The single cell for the from the plants. He can be he is the first person introduced for the PTC can be studied on the single cell only. Okay. The next scientist is Skoog and Miller discovery of a principle of a hormonal control of the organ formation in the tissue culture. Okay. That Skoog and Miller studied on the hormonal control of the organ formation in the tissue culture. Means we are selecting the nutrient medium and the under this nutrient medium that using the different nutrients, the formation of the organ formation in the tissue culture can be studied by the Skoog and the Miller scientist. Then after that the Rennert and the Stewart in the 1958-59 first report on the somatic embryogenesis. Okay, that term as a somatic hybridization can be introduced using the Reinert and the Steve Ward only. Then next study in the 1960 that the Johns, the hanging drop method of the cell culture. Then the Bergman in the 1960, that Bergman cell culture techniques can be introduced, and that cell culture technique is a newly defined by the Bergman, and that itself that cell culture technique called as the Bergman cell culture technique. After that in 1960s that E.C. Cocking scientists isolation of the protoplast by the enzymatic degradation methods can be introduced. Then the as a Morjigi and the Skoog in the 1962 that can be 62 introduced the development of the MS media for the further study of the tissue culture. Then as Goa and the Maheshwari in the 1964, the production of the first haploid plant by the anther using the anther culture techniques can be introduced. Then the using the in the 1917 as a scientist as a power can be studied on the protoplast fusion first. Then next in the 1971 that the Takabi scientists can be studied on the regeneration of the first plant from the protoplast fusion. Then the Carlson in the 1972 first report on the interspecific hybridization through the protoplast fusion. This is all about the plant tissue culture history, that the involvement of the different scientists using the different techniques in the developing the more probable tissue culture that can be we covered here. Thank you so much.